It's my pleasure now to introduce you the last speaker of the OASI Academy for 2025, Professor Raimondi. I hope she's online because I don't see her in the... Okay, Manuela, hi.
Online, can you hear me?
Yes, we do. Good afternoon. But we don't see you. I don't know if you can turn on the camera.
No, we saw her. We see you.
We see. Okay, then it's my problem. So you have to know that I have been inviting Professor Raimondi to be a speaker since January 2025. But she was very busy, and all the time she was busy. She suggested other speakers, and she was all around the world. She was busy, and I kept inviting her. So I was a sort of a stalker of Professor Raimondi, and at the end she said yes, and I'm so pleased that she is the final speaker of our academy, and you will see why. So Professor Raimondi is a professor of bioengineering in Milan at the Politecnico, where she leads the mechanobiology group. She's ranked among the top world 2% scientists. She pioneered the technological miniaturization for the 3 Rs, creating microphysiological system, organoids on chip, implantable imaging window to replace animal testing. Her leadership spans five ERC grants, including 1 consolidator and 1 advanced grant. And she's the first woman in Italy to win an ERC advanced grant in engineering. She has held a visiting position at leading institutions such as the University of Pennsylvania. She's an ERC evaluator. She is author of 370 publications, 10 patents, and she is the founder of a spin-off in Politecnico, where she's president and chief scientific officers. And beside this formal presentation, I have to say she's one of the most inspiring scientists I ever met. So having said this, I leave the floor to Professor Raimondi, and I really thank her for being with us.
Thank you so much, Chiara, for this presentation. It's really lovely. And thank you for organizing this symposium. Can you see my monitor, the presentation?
Yes, we see your monitor. Thank you, Manuela. Good. Perfect.
So congratulations to the organizers of this very important course. And so I will speak about my story. This is something that I hope will inspire the auditors. I am originally a mechanical engineer. I got my 5-year laurea at the Polytechnic University in Milano, but I've been a bioengineer ever since my PhD. So I have a PhD in biomedical engineering obtained in 2000 at the same institution. And I've spent the last 25 years building up my group. My group is the mechanobiology group based at Polytechnic University in Milano at the Department of Chemistry, Materials and Chemical Engineering, Giulio Natta, which is a very important department of Polytechnic University because it's the department where Giulio Natta won a Nobel Prize some years ago. So this is my group now, but in 2005, when I was a postdoc, the group was a lot smaller. As a matter of fact, in those years, I was dreaming about building my lab, my own independent lab. So I didn't have the resources to do this. And the Polytechnic University in Milano in those years didn't have a facility and research lines addressed to, I wouldn't say bioengineering, I would say biological engineering, so biologically oriented research. I was very much interested in mechanobiology, so I was dreaming of building my own lab. And so what you see here is the output of a software that I used in those years to design the ideal lab that I would have liked to have inside the university. I didn't have the resources. I didn't have the equipment. I had 0 resources to build the lab. but so I kept on for years, just drawing and moving the furniture and the equipment inside this CAD, the three-dimensional simulation of a laboratory. So it took me a couple of years to find the resources, but finally, I was in contact. I did research with a Professor Andrea Remuzzi of Mario Negri Institute for Pharmacological Research in Bergamo, and he called me on the phone one day and he said, "Look, we're moving our facilities and we're going to leave some functioning instrumentation that we're going to throw away. And if you want it, you just come and take it." And so this is how the first version of my lab was born. So in 2010, the university gave me the space. And after 1 year, thanks to this, I would say, I wouldn't say grant, but this let's say free instrumentation for the Mario Negri Institute. As you can see in this photograph, my first lab, the first version of my own laboratory of mechanobiology was born. And everything you see in this lab is totally secondhand instrumentation, the safety cabinet, and all the other the tables. Everything is secondhand. So this is how I started doing my research. But also I was very lucky because I started working with a couple of colleagues in physics from my university, like Professor Cerullo. And I started to use a very interesting microstereolithography technique to microfabricate some cell culture substrates. And this technique is called two-photon laser polymerization. It's a microstereolithography technique that allows to build three-dimensional, let's say, forms and structures of any kind of geometry, so with a totally arbitrary geometry with a very high resolution. And as you can see here, these structures are built on a human hair. This is just to give you an idea of the very high resolution of the method. So I started using this technology and becoming familiar with it, this microfabrication technology. And this is how I had my first scientific idea on how to condition the shape of stem cells in order to recreate the structural and physical conditions to these cells to maintain their stemness. So I had this idea to build a microscopic grid scaffold. This is a microscopic scaffold for the culture of stem cells. It is really, really very highly miniaturized because every beam that you see here has a thrust dimension of 1 micron. So it's simply, it's the same scale of single cells. And my idea was to use this three-dimensional adhesion grid to block somewhat the migration possibility of cells, to have them adhering in three-dimensional space at the single cell scale. So I did this with millions of cells. And this condition would keep the mechanobiological transcription factors out from the cell nucleus, as you can see here in the picture. And so this was an idea on which I won my first ERC grant. It was an ERC consolidator grant that I won in 2014. So at this point, I became an associate professor at my institution, also thanks to this ERC grant. And I kept on developing technologies, the nichoid substrate. I call it like nichoid because it sounds like organoid, but it recapitulates specifically the stem cell the stem cell niche, the structural aspects of the stem cell niche. So this is how it became in the years, and also I kept developing other technologies. And one very important result was that I asked for further I submitted further proposals, further funding, because as an engineer, I did research that brought to many patents, and for each patent, I asked for a proof of concept grant that are smaller grants linked to the bigger ones that ERC gives to evaluate the commercial and the technical feasibility of bringing the ideas and the let's say the inventions, bringing new inventions to the market. So I obtained an ERC proof of concept in 2016 for the first time, and this proof of concept was to study the possibility to upscale the production of the nichoid substrate. And we did, especially Tommaso Zandrini, the researcher who worked on this project, he was able to upscale by 10 times the production of these substrates by parallelizing the microfabrication, and also we had a very positive outcome of the commercial feasibility study for the substrate. But in the same years, I also wanted to see inside my cell cultures, I wanted to see not only at the end of the experiments, but also longitudinally in time lapse during the experiments. So I also had the idea of inserting the microscopic scaffold inside a microfluidic cell culture device that I call bioreactor. So I invented, together with my student Matteo Lagana, this device that's called MOAB. It's the millifluidic optically accessible bioreactor. And this is a culture chamber, very similar to the microfluidic chambers. So, but it's millifluidic. It's a little bit bigger in the chamber, but it works like that. You put the cell culture inside a three-dimensional cell scaffold with cell culture inside the chamber, and then you push culture medium through the cell culture in order to and you put the system on the microscope, under the microscope, so you're able to monitor cell culture longitudinally for many weeks in order to follow up on very, very slowly developing biological processes. So this idea won another ERC proof of concept because the device had a very high potential to be brought to the market. Both ideas, the nichoid culture substrate for stem cells and the more millifluidic bioreactors, were very successful in terms of the feasibility of becoming commercial products. So in 2019, I became both a full professor in university and an entrepreneur, in the sense that I have activated and I founded a startup company. It's called MOAB, Miniaturized Optical Accessible Bioreactor. And you see here a presentation of our products. Now these two have become a product that is sold. They are commercially available on the market. And this is my chief executive officer of the company. Also, we had high success with the company in obtaining funds. So the spin-off company was incorporated in 2019, as I said. This is how the millifluidic chamber, the MOAB, looks like now. This is on the microscope, and this is how you can see inside three-dimensional cell populations, for example. Specifically, this was a model of the immune system, of what happens inside lymph nodes, so of innate immunity. We also had very much luck with the grant rising with the company. In 2020, with this company, MOAB, we won a Crack It Challenge, which is a challenge that's funded by National Center for the Replacement, Refinement, and Reduction of Animals in Research. This is a non-profit organization based in the UK that funds research alternative to animal testing and is funded by very big pharma companies. In this specific challenge was funded by Novartis, Bayer, and Takeda. So with all these three companies were interested in finding a way to test the tumorigenicity of gene-edited hematopoietic stem cells. So we set up a very big project funded by 1,200,000 euros for 3 years, with a lot of with a very good partnership with other scientists, in which we developed this model, which is a model with the MOAB of the connection between the bone marrow and the lymph node. to test. This is a model that can run for months, and it's able to visualize and assess the possibility that a cell that is gene edited, and a hematopoietic cell gene edited, becomes tumorigenic. So we did all this work, and we were very successful in obtaining funds also with the company. So my career in university was arrived to full professor, so it was going very well with my group. The company was running very well, but still I wasn't happy of my scientific evolution. I mean, I was happy, but I wanted more. So I wanted to get into in vivo models, because up to then I had developed only in vitro models, and I wanted to develop some models in vivo because I was very interested in adding the microcirculation and also the immune system to my models of physiopathology and of therapy. So I accepted an invitation to move to the University of Pennsylvania in Philadelphia at the Department of Bioengineering, and I was invited by this professor, Professor Gottardi, who had opened this new lab of bioengineering and biomaterials inside the Children's Hospital of Philadelphia. So this invitation was in 2019, and unfortunately, we arranged that for me to move in 2020, of course, before the pandemic came, but I moved anyway. So this was a very heroic decision in my life to move away from Italy by myself, leaving my husband and two children of 13 and 19 years old in Milan during the lockdown in the middle of the coronavirus pandemic. And this is a historical photograph of me that I took when I was on the airplane leaving from Milan to Philadelphia in 15th of September 2020. It was a very difficult decision, but I took it because I really needed 1 year to study and to acquire some new knowledge on the immune system. So I stayed at the Children's Hospital of Philadelphia. This is me working in the lab. I developed a knowledge that I didn't have up to then in the culture of macrophages, because I've never cultured non-adherent cells in my life. This was the first time. And I've learned a lot about the immune system and all these cells and how they work. And when I came back from Philadelphia, I was able to write to get another proof of concept ERC grant, because you can get at maximum 3 from one main ERC grant. So I got the third one in 2022. And this idea was coming back from 1 year working in a children's hospital, was to use the nichoid substrate to culture mesenchymal stem cells and to produce a secretome from these cells that would be able to treat fibrosis in this condition that's called child lung disease. So it's a fibrotic, it's progressive fibrosis that affects children, and we did this very important project, and we arrived at the end with good results in terms of a product that can be used to treat interstitial lung disease in children, but also in adults. So this was my 4th ERC grant. So having won 4 ERC grants, when I came back to Italy, I set up a non-profit organization. It's called ERC in Italy. This organization is to help Italian scientists and young scientists mostly in acquiring more grantsmanship, in getting more ERC grants. And you see here that I'm now the president of this association, and I'm here with my vice president and my delegate for international relationship with the government, and we do a lot of tutoring, both to groups and to single people. We put ERC grantees in contact in Italy between each other, Italian ones, of course. And also we transfer in Italy the best practice of the ERC executive agency to in the generation of grant calls and in the evaluation of the proposals. And also we do some sensibilize what's called outreach. So we try to explain to the public how their money that they pay in taxes is used for research. So this is my third work that I do. So I work in university. I am a president and chief scientific officer of the spin-off company, and I'm the president of this nonprofit association. So what's next? Well, the next has already begun. When I came back from the United States, I had acquired more knowledge on how the microvascularization and the immune system was working and how it would influence the process called fibrosis. So on the fibrosis progression, on fibrotic progression. So my interest was in developing a model, an experimental model of fibrosis, which is not at all present and it's not available to study fibrosis. So I had this idea to somewhat incorporate a microfluidic culture chamber like the MOAB that I've shown you, holding a model, a cell model inside made of human cells, possibly, to incorporate this with an entire organism so that this entire organism could, with its blood vessels, revascularize all the cells. So this this model of fibrosis made with human cells and create a fibrotic reaction. This whole organism that we use is not an animal. Technically, it's an embryonated avian egg, and the embryo is not considered a living animal up to a certain development stage because it cannot feel pain, okay, until day 14. So this is true for all embryonic organisms. So it's a lot more ethical than using adult animals. So I had this idea to create a hybrid model, which was a hybrid between an in vitro model that is very much controlled and is based with on human cells, so it's humanized, and an in vivo model, which could provide vascularization. As you can see here, these are the blood vessels of the embryo, of the chorioallantoic membrane, a breathing structure of the embryo. And you see in the collagen of the fibrotic reaction recreated in my microenvironment in vitro. And the system can be put on the microscopic objective, so it's highly inspectable. This idea valued me an ERC advanced grant that I won in 2021. That is now, it started in 2022. It's now ongoing. And this is one I wanted to show you, the first preliminary result of this project. Of course, we're still at the half of it, so we're still working on this hybrid in vitro, in vivo model of progression. The title of the project may interest you very much because the title is Mechanobiology of Cancer Progression. This is because these fibrotic microenvironments I'm interested in are fibrotic microenvironments caused by solid tumors, specifically the reason for the drop in the progressive fibrosis in solid cancers. Of course, everybody knows that is causing a drop in efficacy of chemotherapeutic agents. So I wanted to create a model able to be used to test new cocktails of drugs to oppose, to limit this progressive drop in efficacy of chemotherapeutic agents. So this is the first good output of the model. You can see the red blood cells flowing in the microvascularization of a fibrotic microenvironment recreated using human cells. In green, you see the human cells. These are cancer cells specifically. And what you see in blue, and this is all autofluorescence, so it's not labeled. It's 2-photon microscopy, so you don't even need labels. And in blue, you see fibrosis. You see the collagen fibrils that are created by the cancer-associated fibroblasts. So this is a very good result in terms of imaging and high resolution. So now, as I said, my lab is involved in this very big ERC advanced project. And this is a picture taken in the lab last year, as where you can see now, the lab, the mechanobiology lab, has now become halfway between cell culture lab and hen house, because we're always working with this chicken embryonated chicken egg models, and we're still developing technologies to, let's say, to improve our knowledge in this field. So I've come to the end of my presentation. I hope I still have a couple of minutes just to thank all the people, this very beautiful scientists that are those with whom I've partnered in these years to develop all my projects. My take-home message to everybody who is listening is when you're able to win the competition for competitive grants, then my suggestion is to start asking for new competitive grants, because you're a lot more competitive when you have already won some of these grants, so you will be a lot more successful. This is what I've did and what I'm still doing right now. And I would like to thank also the people from my group that are very beautiful scientists. And I thank again all my funding bodies, and these are all my the lab website, the company website, the ERC Association website, if you're interested in other information on my activities, and if you want to contact me, this is my email, please. Thank you so much for your attention.
Thank you, Manuela, really thanks. So I leave the audience writing questions for you in the chat. And I want to start with a couple of questions for you. So Manuela, we have an audience of physician scientists in the field of medicine, oncology. And you know, quite often physician, and I'm a physician, so I can say this, we stuck into our small box of things to do, of clinical outcomes, of drugs, dosage, etc. Okay, and we stick here. However, innovation is made when we have interdisciplinarity, when we break barriers of our field. And this is also a sort of requirements when you present an ERC to show that you are thinking out from the box. So what is amazing for you in your story is that you started as a mechanical engineer. And you broke the barriers of engineering, of biology. Now you are in the field of cancer. You are breaking the barriers of medicine, even clinical medicine. So what is your advice for junior investigator? How to break barriers?
I think the best advice is to grow up with this kind of mentality. So the best advice to be, if you like frontier science and you're open to the idea of being open to other disciplines, multidisciplinary, I think the best thing is to find a mentor who has already won one of these grants, because it's very important to learn the mentality. Your mentor is the person. Your scientific mentor, I mean, is the person who is going to teach you the approach. So if you want to study and to develop a multidisciplinary or transdisciplinary approach, which is my approach is more than multidisciplinary. So I am an engineer who works in biology since 25 years. So I've changed totally my field of interest. So this is transdisciplinary. So if you want to have this kind of mentality, it's very important that you grow up in a group where this is the norm, where this is normality, and then normality will become your normality. And in this regard, I had this year the most important success in my career is that my younger collaborator has won an ERC grant. So he has become independent, and he has been successful in applying his curiosity to a different field from mine, and he won his independent grant. So I think it's infectious. I think the transdisciplinary mentality is infectious. So go and work in a group where you can be infected. This is my advice.
Very good advice, Manuela. Thank you. Well, I see there is a question in the chat. Probably they are referring to what you show with neonates affected by lung fibrosis, interstitial lung disease. And they say, how did you involve patients in your research? Probably.
Yes, I partnered with the Buzzi Hospital in Milano, where there is one surgeon that who is my partner in the project. But the project is a proof of concept, an ERC proof of concept. So it's not a clinical project, a clinical grant. So the aim of this proof of concept grant was to obtain a commercial feasibility of selling a product based on secretome. And this is done by consultants in business development, and also the technical feasibility. And the technical feasibility was studied in vitro. So the ERC proof of concept grants are really providing you just with the proof of concept that you can go on the market with such a project. The proof of concept was there, is given. So now we're applying to further grants that hopefully will bring the product to all the way to the patient, and that means to the market. But we need to ask for other grants and involve patients. We did not involve patients at this in this first grant.
Okay, thank you, Manuela. Another question for you, very practical, very practical. So to me, it's also amazing that you were designing, like an interior designer, your lab before actually having money to do the lab. And we always think the contrary. First, I get the money, and then I do the interior design of my lab. So you started really planning and having the vision of what would have been possible and what was your dream. And this reminds me, you know, when I read about the two tennis players, Serena and Venus Williams, the father started planning the life of the daughters 20 years before. So you did the same. And here is the question for you. Talking about timing for a junior investigators, for instance, a junior investigators that just got the PhD. Okay, thinking about the timing, planning, and of course, visioning. What is your advice in organizing the writing of the ERC, in getting collaborators, in putting together things and planning?
I think that if you already have your PhD, you're already late. So in the rest of Europe, during your PhD, you start asking for travel grants if you want to travel to conferences. You start asking for any type of grants, like intramural grants if you want equipment to do some different research. So the decision I think you should take very, very early in your career is if you want to do science that is frontier science or incremental science. This is the point. Okay, incremental science means science done on some research lines that are already available. Okay, like I already developed a protein, I optimize characterization of this protein, for example. This is incremental science. It can be of very high quality, and the European Commission funds by 80% by 90% of the budget incremental science. And if you want to do this, and if your personality is more oriented on this type of science, it's good to work in a group that has a lot of Horizon 2020, Horizon Europe funding, and they work with collaborative research in collaborative big consortia. So you learn this kind of mentality. Instead, if you want to do frontier research, which the European Commission funds with the ERC by only 10% of the budget, of course, the new frontiers are opened by 10% of the people. Cannot be everyone work on new frontiers. You need someone also consolidating the existing lines, okay? But if you want to open new lines, then as I already said, the important thing is to work, for example, your PhD should be done in a group where one PI is an ERC granted PI. For one practical reason, because they have money to pay for your fellowship. You have a higher probability that they have the money to set up your fellowship, but also because they will transfer you the mentality of frontier science. But this is this is very difficult because it means that you are independent. You want to be independent as soon as possible. So you have to start asking grants and writing grant proposal during your PhD. And this is news also for the ERC. You can ask an ERC grant now, the next call, starting from the date 0 of your PhD. You don't even have to wait for 2 years. Okay, so you have to learn how to do this during your PhD, and you will become an independent researcher, an independent scientist, and you will be totally independent, but you need total independence on your funds. This is the reason why I designed with the interior design software, my lab, because my group said, look, what you are doing is not of interest with respect to our existing research lines. Okay, we do other stuff, biomechanics, cardiovascular biomechanics, more incremental research in biomedical engineering. So this is frontier research, mechanobiology. So it's okay for us, but you have to be 100% independent. So we're not going to fund any of this research. So you have to be independent. So my effort was for desperation, was not just vision. Okay, there was good vision that was going to become important, this kind of research, this kind of research line and technologies, but it was also desperation. So if you're in Italy, this is very common, that there is no money for research from the ministry. So you have to learn as soon as possible to become very good in grant writing and to do this as much as possible. But just to recapitulate all my very long answer, the big decision is, do you want to do incremental science and become high quality scientist in this field, or do you want to do frontier science and become a high quality scientist in this other kind of approach? So are you more team player, or are you more a high achiever? What is your personality? If you are a high achiever, usually you are not much a team player. It's the ERC is good for you, independent research, frontier research. If you're more a team player, then go towards incremental research with all the European funding, which is, as I said, 90% of the budget is on this kind of funding schemes. So it's also more easy to catch.
Okay,
I hope I was able to answer.
I really like your psychology test. Are you a team member, a team worker or not? Yes. Okay. So, final question, Manuela. Because we spend the whole year with our webinars, residential course, talking about money, grant, publication, so how to become a superhero in science. Okay. So I really thank you that you said it was desperation. It was not it was not vision, but it was desperation. But you made it. You reached a very high level of success. A more personal question, just because you mentioned this and just because we talk about this. You are a superhero, super scientist, but also you are a wife and a mother. And you decided to move to the US during the pandemic, so in a special moment for families. So you are showing also that it's possible to be a superhero in science and to be a mother and a wife. How to do? Do you have any advice to our audience? How to find the balance, professional life as a professor, president, CEO, founder of startup, and the personal life?
It's not possible. It's not possible to find a balance. So you will always be unhappy When you work a lot, you miss your children and your husband. When you are with your family, you miss your work. So you will have different epochs and periods in your life in which you put more energy on your family when your children are very young and they're very small. Then there are other periods in which you put more energy in your work when your children are grown-ups, for example. So I don't think there is a balance. There cannot be. But the best advice I can give to young women, young scientists, female scientists, but also men, I think. But to women, my advice is to find a good partner. So the only possibility to make it in finding a balance, which will be a dynamical balance, which will change during your life, is to find a good, I wouldn't say husband, but partner, a good partner, someone that values you and that respects the ideals that you are pushing ahead in doing scientific research. And this is very, very true for women. It's also true for men.
Well, Manuela, I would conclude here. Really, thank you so much for sharing everything, your vision, your life, where your heart is. And I'm so happy that I have been stalking you. So now you finally gave this webinar. Thank you again, really. I will come soon to visit you in Milan because I need some of your positive energy. So
please
thank you. Thank you, Manuela, again. And now it's the time for the final message from our OSCI board member and member of the Scientific Committee of the Academy. So I leave the floor to Professor Sandstrom for a few words of conclusion.